Good morning, everyone. Let's go ahead and get started. I have a couple of announcements briefly before we begin. First is that, as hopefully all of you have received the B-Space message, the homework on genetics, which should be a pretty simple one, is available now. You can download it from B-Space, or we'll have hard copies that you can get from Brad. It will be due this coming Wednesday, the 25th, at 12 p.m., either in class or as an email attachment to your GSI. Also, just one time only, because today Yvonne Kim is unable to be here, Brad Dotson is going to hold the discussion section here in this room. So he'll have more space than the GPB rooms for the regular discussion section. So if you want to come to discussion, if you're in either of the sections, please just stay here afterward and attend with Brad. And he's made a nice handout for more genetics practice. So are there questions from Wednesday's lecture? No? Great. Okay. We're going to carry on then. What we learned previously is that test crosses are a very important genetic tool that allows us to determine the genotype of an individual parent with a particular trait that you're interested in studying based on the phenotypic ratios of the progeny, the offspring of that parental individual. And we've learned that you can cross your parental plant to a tester line that you already know is homozygous recessive for the trait or traits that you're interested in, whether it's flower color or seed shape or plant height. You then analyze the phenotypes of the progeny using the Punnett squares and determine the phenotypic ratio. So here's another example of a more complicated situation where you have a dihybrid test cross where you're looking at two traits of interest, in this case looking at seed color and also seed shape. And so these are three potential genotypes of individual plants where the phenotype is the seed is yellow and round. But you could have different combinations of alleles, as we saw on Wednesday, to produce those round yellow individuals, because yellow and round, as we already have learned, are dominant crosses or dominant phenotypes. So we can go through each test cross, and what you'll see is that when you cross the parental plant of interest to your test crosser, which you know is recessive at both the seed color locus and the seed shape locus, depending on the genotype of the parent of interest, you're going to get a different outcome. So this is what makes it so valuable. In the situation where you had a parent that was homozygous dominant for the yellow allele and heterozygous for the round allele, this individual, when crossed to your heterozygous recessive test or your homozygous recessive tester, is going to produce two different combinations of gametes, right? It's either going to produce big G, big W, or big G, little w, and that's shown here. The recessive line is only going to produce the gametes little g, little w. So you do a very simple Punnett square, where you have, in this combination of alleles, big G, big W, little w, a double heterozygote. And in this combination, you're going to have big G, little w, little w. So when you look at the phenotypes of these plants, then 50% are going to have this genotype, and they're going to be yellow and round. Whereas 50% are going to have this genotype, and they're going to be yellow and wrinkled. So that's one possible outcome from your test cross. If, however, your original parental individual was heterozygous at both loci, then you're going to get a different outcome from your test cross. In this case, this doubly heterozygous individual is going to produce 4 different combinations of alleles in the gametes. It can either produce big G, big W, big G, little g, big W, or little g, little w. So when that's combined with the alleles from the test parent, you're going to have 4 different combinations of outcomes and 4 different types of progeny, where 1/4 are going to be heterozygous at both loci, 1/4 are going to be heterozygous at the yellow locus, homozygous recessive at the round locus, 1/4 they're going to be homozygous recessive at the yellow locus, heterozygous at the round locus, and then 1/4 that are going to be doubly homozygous recessive. So the phenotypes are going to come out differently. Here you're going to have 1/4 that are yellow and round, 1/4 that are yellow and wrinkled, 1/4 that are green and round, 1/4 that are green and wrinkled. A third combination is shown here, and I won't bother to go through the details. It's essentially the same process as what we've done for the other two crosses. And you'll see that there's one combination of alleles that's actually not up here. And you can go ahead and do this with a Punnett square on your own. And that's a combination where you have big G, big W, crossed to your tester line. But the bottom line is that these test crosses illustrate what combination of alleles you have, because each different combination of alleles gives you a different outcome in the progeny. So is that clear to everyone? Great. So I want to spend a little bit of time going back and talking about probability. We've already looked at different ratios of events in terms of alleles and how they're incorporated into F1 and F2 generations. But I want to spend a little bit more time on probability because it's a very powerful application for genetics. And just to remind you, the definition of the probability of an event is the chance that the event is going to occur in the future. We can define probability as the number of times an event would occur divided by the total number of possible events. As a result of this equation, probabilities are going to range from 0, an event is absolutely certain not to happen, to 1.0, an event is absolutely certain to happen. And we usually give the range in between as a percentage. All right, so the simplest example is a coin flip. The probability of heads in a coin flip is the probability of the number of times an event is going to occur. You're going to do one flip, so it's 1 heads, divided by the total number of possible events. Well, you could have heads or you could have tails. So 1 heads plus 1 tails. That gives you 1 divided by 2, which is, of course, equal to 0.5 or 50%. So flip a coin, you have a 50% probability of it landing heads. How this is important for genetics is that there is a product law that allows us to easily discover simultaneous outcomes. So if we're talking about 2 traits, seed shape, seed size, and we want to know what the probability is of obtaining both of these traits in certain combinations, we can easily work out those probabilities based on product law. So again, to use a simple example, what's the chance of rolling snake eyes, which is just rolling 1, obviously, with 2 separate dice? The chance of rolling 1 with the first die is 1. Obviously, that's only one side, out of a possible total number of events, which is 6, because there are 6 sides to the die. The chance of rolling 1 with the second die, because it's exactly identical to the first die, is the same. So 1/6 probability of rolling 1 with the first die, rolling 1 with the second die has the same probability. So the chance of rolling 2 1s with both of the dice is simply 1/6 times 1/6. 1 times 1 is 1, 6 times 6, 36. So the overall probability of having outcome 1, rolling 1 with the first die, and outcome 2, rolling 1 with the second die, is 1 in 36. So if you were to roll these 2 dice 36 times, the probability is snake eyes would come up once. So we can, as I said, use this product law when calculating probabilities of inheriting different combinations of alleles. And this gets really important when you start to do crosses where you're looking at multiple traits of interest. For example, in breeding, where you would want to breed into a plant disease resistance and increased yield and drought resistance. You've now got a large number of allelic combinations to work with. So how this probability can be applied is using something called the forked line probability method, and it's not as scary as it sounds. It's just a simple tool for analyzing dihybrid or trihybrid cross phenotypes. So here's an example in which you're taking a doubly heterozygous plant that phenotypically is yellow, but is heterozygous at the yellow locus, and phenotypically is round, but is heterozygous at the round locus, and crossing it to itself. In the F2 generation, if you look at all of the offspring, just observe their phenotypes, because the yellow allele, big G, is dominant to the recessive little g allele, we can say for trait 1, the yellow trait, 3/4 are going to be yellow, right? Because they're either going to get big G, big G, big G, little g, or big G, little g. So 3/4 are going to be yellow, 1/4 are going to be green. Looking at the second trait, the seed shape trait, we can say out of this 3/4 of the offspring that are yellow, 3/4 of those are also going to be round, because the round allele, big W, is dominant to the wrinkled allele, little w. 1/4 are going to be wrinkled. In the same way, of this 1/4 of plants where you have green seeds, 3/4 of those are also going to be round, and 1/4 are going to be wrinkled. So then you can simply multiply the probabilities together for each of the potential allelic combinations and very quickly arrive at your answer. So what proportion of F2 offspring are going to be both yellow and round? Well, we know that 3/4 are going to be yellow, 3/4 are going to be round. So 3/4 times 3/4, 9/16. What proportion are going to be yellow and wrinkled? Well, we know that 3/4 are going to be yellow, but only 1/4 of those are also going to be wrinkled. So 3/4 times 1/4, 3/16 are going to be yellow and wrinkled. For the 1/4 that are green, how many are also going to be round? It's going to be 1/4 times 3/4, 3/16. And what proportion are going to be doubly recessive, being both green and wrinkled? 1/4 times 1/4 is 1/16. It's a very easy way to determine the probability of getting any particular combination of traits. So let's look at a more complicated scenario than a trihybrid cross. Now we have 3 different loci generating 3 distinct traits. In your parental generation, let's assume that the first parent is homozygous dominant at all 3 of those loci, and you're going to cross it to a tester plant that's homozygous recessive at all 3 of those loci. So the gametes of this parent, the only combination is big A, big B, big C. In the gametes of this parent, the only combination is little a, little b, little c. So the only combination in the F1 is going to be a plant that is heterozygous at each of the 3 loci. Right? That's very straightforward, I hope, by now. Okay, but now let's cross this plant to itself. Well, now you have 8 different combinations of alleles that can go into the gametes. You can have big A, big B, big C, big A with big B and little c, little a with big B, big C, et cetera, et cetera. So now when you do a Punnett square, you're going to have to put all 8 of these gamete combinations along one side of the square, plus all 8 along the other side of the square. And you're going to end up with an enormous Punnett square with 64 squares in it, which is great, and I recommend that you do this. But you can also cheat a little bit and use that fork-line probability method to figure out what the phenotypic ratios are in the F2 of the cross. And we do this in the same way that you would do it for a dihybrid cross. For the first trait, if A is dominant, then 3/4 of the individual progeny are going to be big A. They're going to have whatever the A trait produces. Among this 3, and then 1/4, of course, are going to be small a. They're going to be little a, little a. Among this 3/4 that have the big A phenotype, 3/4 are also going to have the big B phenotype, because that's also dominant. 1/4 are going to have the little b phenotype, because that's recessive. So among this 3/4 that has the big B phenotype, 3/4 of those are also going to have the big C phenotype, because that's dominant. 1/4 is going to have the little c phenotype, because that's recessive. For the 1/4 that have the small b phenotype as well as the large A phenotype, 3/4 of them are going to have the big C phenotype, because that's dominant. 1/4 are going to have the little c phenotype, because that's recessive. So then if we want to know what proportion of those 64 plants are going to have the big A, big B, big C traits, we simply multiply the probabilities together. 3/4 times 3/4 times 3/4, 3 times 3 times 3, 27, 4 times 4 times 4, 64. So 27/64 of those progeny are going to be big A, big B, big C phenotype. Now, remember, they're going to have slightly different genotypes because some of them will be homozygous at each allele, some of them are going to be heterozygous at each allele. Yes?
Are heterozygous in an allele and they're homozygous recessive or homozygous dominant? Can we still, so like if it was not trihybrid but many more genes, is it possible to just do a Punnett square for the gene where you're homozygous dominant or recessive with the other parent and just use this relationship except change the number for the homozygous dominant to like 1/2 or whatever it's going to be?
Yes, yes. Yeah, I'm just showing you the combination where you have the most different types of probability to show you how much you can streamline this. But yes, you can use other parental combinations with this same system. Yeah. So for example, if you want to know what proportion of your F2 progeny plants are going to have a small a phenotype, a big B phenotype, and a small c phenotype, you would go back to your 1/4 of plants that have the small a phenotype. Of those, 3/4 are going to have the big B phenotype. And among those, 1/4 will have the small c phenotype. So now you take 1/4 times 3/4 times 1/4, and the outcome is 3 out of 64. How many plants are going to be triply recessive for all 3 alleles? 1/4 times 1/4 times 1/4. Only 1 out of every 64 plants you're going to examine is going to have the small a, small b, small c, triply homozygous recessive phenotype. So all the different allelic combinations come out to 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1 ratio. And that adds up to 64. So I want to move on now and briefly touch on the principle of independent assortment, which hopefully is familiar to most of you. And that principle, as described by Mendel, asserts that segregating alleles assort independently during gamete formation. So one is not dependent on the presence of another. The segregation of any 2 alleles for one gene has no effect on the segregation of 2 alleles at another gene, with one major exception that I'm going to come back to. So just an example of this, the assortment of yellow and green alleles for the trait of seed color has no effect on the assortment of wrinkled and green alleles for the trait of seed shape, and vice versa. And this is, again, really important advance at the time, because that was another nail in the coffin of this whole blending idea. In the blending idea, things would have to segregate together in order to produce an intermediate combination. But what independent assortment allows is extensive genetic variation that we actually observe in the world in a phenotype of birds, in a population of birds or a population of people. And that's because the number of possible different gametes that can be produced by any set of parents is equivalent to 2 to the power of n, where n is the haploid chromosome number. So there are 10 haploid chromosomes in Arabidopsis, for example. So the number of different gamete combinations is 2 to the power of 10. Now, why do I introduce chromosomes here? That's because observations by several geneticists, William Bateson and our friend Reggie Punnett here, in the early 1900s showed that not all genes assort independently. And of course, this seems pretty obvious to us because we know that genes are found on chromosomes. So really, it's only genes on different chromosomes or very, very far apart on the same chromosome that are going to assort independently. So this is the exception. We know that chromosomes contain thousands and thousands of genes, and they don't assort independently because they're physically located near one another. And linked loci, therefore, tend to be inherited together. So the closer 2 genes are physically to one another, the more likely they are to be inherited together. So this is just a schematic of how linkage prevents independent assortment. So here's a diploid cell after the chromosomes have replicated. Here is one chromosome with the 2 sister chromatids, and here's the second chromosome from the other parent with the 2 sister chromatids. And in this case, we have homozygous dominant alleles. Here we have homozygous recessive alleles being provided by each of the parents. As this cell undergoes meiosis, all the gametes separate. So now this sister chromatid goes into this gamete. This sister chromatid goes into that gamete. This sister chromatid into this gamete. This sister chromatid into this gamete. But we see that whatever combination of A and B alleles came from the parents are produced are replicated in the progeny. So big A from this parent stays with big B. Little a stays with little b. So loci A and B are linked on the same chromosome and will usually be inherited together because of the process of meiosis. But of course we know that there's a big exception to that as well. And that's because crossing over occurs. And we know that phenomenon is recombination, which occurs during prophase 1 of meiosis in all cells. And that, of course, is where non-sister chromatids, so chromatids from the two parents, from the homologous chromosomes, exchange DNA segments. And it takes place at random locations. What that affords, then, is the production of novel recombinant phenotypes. And that's why none of us look exactly the same as our parents. So again, to show this diagrammatically, the exchange of genetic material between chromatids, this recombination activity, occurs during meiosis. And what happens is you have the homologous chromosomes pairing with one another, right? You remember that from Bio 1a. And here you have the two sister chromatids from the first parent, which have big A and big B at the two different loci. Here are the two sister chromatids from the second parent, which have the genotype of little a, little b at the two loci. During the process of meiosis, some of the chromosomes, some of the chromatids do not cross over. So they're inherited here and here in a non-recombinant way. And unfortunately, they switched the colors on this scheme. So big A, big B is actually supposed to be the blue chromosome, and little a, little b is supposed to be the purple chromosome. So don't get confused about that. Just goes to show you that even good textbooks still have occasional mistakes in them. But the point is that you do get crossing over at some points on sister chromatids. And what that is literally an exchange of genetic information, where this part of the chromosome then exchanges its end with the one over here, and vice versa. It's an equal exchange, 1 to 1. So then you have recombinant alleles in these two gametes, where this gamete has inherited most of the purple chromosome, but now part of the blue chromosome. And this other gamete has inherited most of the blue chromosome and this little part of the purple chromosome. And that's now changed the combination of alleles. Instead of big A going with big B, you have little a going with big B, and big A going with little b, right? So recombination reassorts even alleles that are linked together physically into new combinations. We can actually measure this frequency of recombination between two genes because it's proportional to the distance apart physically on the chromosomes. So two genes on the same chromosome are considered to be linked to one another, physically close to one another, if the recombination frequency between them is low, less than 50%. The more closely linked the two genes are, the less likely it is that a recombination event will occur between them. And thus, you get a lower recombination frequency. And some recombination frequencies are less than 1%, very, very low probability events. And so if you can think about this, imagine, it's basically the same way that, imagine genes on a chromosome being like houses in a neighborhood. So the probability that the city would build a freeway between your house and the house at the end of the street is pretty low because they're physically so close together. But the probability that they might build a freeway between your house and a house 2 miles away is considerably larger because there's a greater distance between them. Does that make sense to everyone? It's exactly the same principle as what we're seeing here. So we can use this recombination frequency as a tool to determine the order of genes on a chromosome and also the relative distances between them. And this is still very useful today in plants where the genome, the exact order of genes on the chromosome, has not been discovered. And that's still true for many, many different plants. So you still have to use this technique in order to find out what combination of genes occurs at any given place. So the method for doing this is gene mapping. And it uses test crosses to identify recombination events among the progeny of the cross. And then determining the number of recombination events among the total number of progeny is going to give us the recombination frequency. We use this as a percentage. So 1% recombination frequency, we then consider equal to 1 map unit of distance between the two genes, because the map units on a chromosome are arbitrary. This measurement is highly accurate for short distances between two genes. It's less accurate for slightly longer distances, and I'll show you why. And 1 map unit has been renamed 1 centimorgan after the very famous geneticist Thomas Hunt Morgan, who was one of the pioneering fly geneticists at Columbia University and was one of the first people to establish how this could be used for mapping different order of gene combinations. So let's map the distance between gene A and gene B on a chromosome. In order to do this, we'll start with 2 parents, 2 pure breeding lines. One parent A is homozygous dominant for locus A, homozygous dominant for locus B. We cross that to our tester, which is homozygous recessive at both of those loci. So there's only one possible arrangement of gametes that can come from each one. Big A, big B from this parent, little a, little b from this parent. So these 2 alleles are going to come together in the progeny. And then we're going to cross these F1 plants back to the tester, which is, again, doubly recessive, little a, little b. And then we're going to look at the F2 progeny of this test cross. And what we're going to observe is different outcomes, where you have different combinations of alleles. You're going to get the parental alleles back, where you have big A, big B, and it's now crossed to little a, little b. Right, so this is big A, big B, little a, little b. Little a, little b from here is also going to assort with little a, little b from your tester. So these are the parental genotypes. They're the same alleles in the same combination as what you had in the F1. All right? But there's actually going to be more than a quarter of these. However, there are also going to be some new combinations that we hadn't seen in either of the parents. And those are examples where a recombination event has occurred between locus A and locus B. And I've just marked that with an X there. So there are going to be some individuals in the F2 that got the big A portion of this chromosome, but then a recombination event occurred. So they got the little b portion of that chromosome. So this is not a parental combination. The parental combinations were only big A, big B, and little a, little b. Now we're getting a big A, or a big A, little b, little a, little b. You also get a combination where this part of the chromosome, little a, crosses over to this part of the other chromosome with big B. So now you have a combination where you have little a, big B, and little a, little b. Usually these recombinants are going to be less frequent than the parental type if linkage occurs. So the first thing to do is simply to note the number of each of these allelic combinations and add them all up to get the total number of progeny. Okay, that's the bottom part of your probability calculation. The top part of your probability calculation is what is the recombinant numbers. In this case, it's 132 plus 136. The total there is 248. So you have 268 recombinant events out of a total of 1,448 progeny. That equals 0.185 recombinants per progeny, or 18.5% recombination frequency, which we express then as 18.5 centimorgans. So can you then say that allele A is linked to allele B on the chromosomes? Anybody? It is, yes. They are linked because the recombination frequency is less than 50%. So now we can take this a step further and map and order 3 different genes. So again, we're going to set this up in the same way. We're going to start with homozygous lines. We're going to cross parent A or parent 1, which is homozygous for big A, big B, and big C, little c. And now note that I've written them in the order A, B, C. But that's not necessarily the order in which they are on the chromosome. It's just alphabetical order to start with. Okay, so keep that in mind. And we're going to cross this parent with our tester line, which is recessive at all three of these loci. Little a, little b, little c, little c. Okay? So the parental chromosomes inherited by the F1 progeny are going to be big A, big B, big C from this parent, little a, little b, little c from that parent. So we're going to cross these F1 progeny back to our tester line, little a, little b, little c, and make observations on what combination of phenotypes you see in the F2. So in this example, in the F2, you see 580 plants that have the big A trait, the big B trait, and the big C trait. You have 592 that have the little a, little b, little c trait. Now those are the same traits as the parents had, right? But you also find all these other different trait combinations. You have 3 guys that are big A, big B, but little c. That's not a combination we found in the parents. There must have been a recombination event that took place there to recombine the big A and big B alleles with the little c allele. We had 5 plants that were little a, little b, and big C, another recombinant that you didn't find in the parents. Same down here, you have big A, little b, big C, big A, little b, little c, big A, or little a, big B, big C, little a, big B, little c. So again, we have a combination of 1,448 progeny, but now we've got a different combination of recombinants. So let's look first at A and B. Then we'll look at B and C, and then A and C, and figure out what the linkage is between locus A and locus B. So between locus A and locus B, the recombinants, not the parentals, remember we're excluding the parentals now, the recombinants that have big A and little b here, big A, little b, 45, but also big A and little b here, 89. What about little a and big B? Little a and big B here, 94, and also here, 40. So we add all of those recombinants up. We get 268 out of a total of 1,448, 0.185. So the map distance between A and B is 18.5 centimorgans. What about the distance between locus B and locus C? Our recombinants are going to be big B, little c, and big B, and little b, big C. So big B, little c, there are 3 here. Big B, little c, 40 there. Little b, big C, 5 here, and 45 there. So we add up all these recombinants, we get 93 out of a total of 1,448, which is 0.064, or 6.4 centimorgans. So is locus B closer to locus A or to locus C? It's closer to C because it's a smaller distance, map units between them. The third possible combination is between A and C. Our recombinant alleles are going to be big A, little c, or little a, big C. So big A, little c, here are 3. Big A, little c, 89. Little a, big C, 5. Little a, big C, 94. We add all of those together, get 91 recombinants out of 1,448, 0.132. So the map distance between A and C is 13.2. So then we can order the 3 genes on the chromosome. The order has to be A, 13.2 centimorgans to C, 6.4 centimorgans to B. The distance between A and B is bigger than either the distance between A and C or the distance between B and C. The distance between A and B is 18.5 centimorgans. Okay, does everybody follow how we got that? All right. But there's a little problem that even I with my math skills can figure out, which is how come 3.2 and 6.4 doesn't equal 18.5? our map doesn't quite add up. The reason for that is because we've not explored the possibility that there can be two recombination events. Recombination events between A and C, and recombination events between B and C. We haven't accounted for those yet. So A and B are the furthest apart. So there can be more than one recombination event. So when we look for single recombination events, we got 45 plus 89, 45, 89, 94, and 40. That's where we have big A, little b, or little a, big B. But there are other recombinants, and that's these guys, where you have big A, big B, but little c. Now, C is in between A and B, so that's not a parental combination. You never got big A, big C, big B in the parents. So that has to have been a recombinant, but it has to have been a recombinant on both sides. So let me just draw that. A, C, B. So here's the chromosome where you have big A. It's crossed over here to little c, and then crossed over here again to big B. So this is one recombination event. This is two recombination events. Now, obviously, two recombination events are going to be less frequent than one. And that's consistent with the numbers that we see. It's the smallest populations that are these double recombinant events. But we have to add those into our calculation. So we go back to our A and B calculation. And we've got 45 plus 89 for that combination, 94 and 40 for that combination, plus our double recombinants, 3 plus 5. But remember, we're measuring every recombination event. So 3 plus 5 is one recombination event in each of those individuals. But there were double recombinants, so there were two recombination events in each of those 8 individuals. So it's a total of 16 recombinant events that we're adding. Okay, is everybody clear on how we got that? All right. So adding all of these numbers together, we now have a total of 284 recombination events out of our 1,448 total progeny. That gives us a recombination frequency of 0.196, or 19.6 centimorgans. And I think we'll find if we add 13.2 and 6.4, we get 19.6. So now we've got all the facts lined up. So the longer the distance between the 3 different loci on a chromosome, the more double recombinants are going to occur, and thus the greater potential to underestimate the recombination frequency. So that's something you always have to factor into your calculations when you're making these larger scale map orders. So what I've hoped to communicate to you in these past couple of lectures is that genetics is an extremely powerful tool. It's a way in which we can determine the inheritance of traits in a laboratory situation or in the field. It's a way to breed traits using quantitative methods or qualitative methods. Qualitative methods be just looking at the offspring. Quantitative methods being applying the forked line probability method. It's a way to analyze new traits that are generated using, for example, mutagenesis or transposons, and we're going to talk about those later in the course. It's also a way to identify specific genes that are associated with traits using modern sequencing, genomics, and phylogenetic studies that Bob Fisher is going to be introducing next week. And it's also a way to associate genes with traits using reverse genetics and transgenic technology that we're also going to discuss in future lectures. But I'd like to close now by talking about some actual real research applications for genetics. And one of the things that geneticists in a lot of laboratories do is try and identify new genes that are involved in making a certain trait. So in this case, we're interested in a trait where the flowers of Arabidopsis, which is the lab weed that's used in many laboratories, we're looking for a trait in which it makes more flower organs. And you can see that compared to wild-type Arabidopsis plants, which always have 4 petals, we can identify variants of that have more petals, 5 or in this case even 6. The way we can identify these is by doing a mutagenesis, and again, we'll talk about this later on in the course, but this sign just means mutagenized population. So now we have a plant that has a different phenotype than the wild type for this trait. So we call it extrafloral organs. We cross this extrafloral organs plant to itself, make a pure breeding line. If you cross EFO plant 1 to EFO plant 2, you get all EFO plant progeny. So now we can ask, is EFO dominant or recessive? So how would we do that? What sort of experiment would we do? Any thoughts? Yeah.
You do a test cross.
Right, and what would you cross to?
You would take the F1 and cross it to a homozygous recessive.
Well, in this case, we're going to cross it to wild type, actually, because this is already homozygous recessive. Okay, but you're definitely, the test cross was exactly right. Here we're going to cross our homozygous recessive EFO plant to wild type, which is big EFO at both loci. And why do we use 3 letters? In all of the other genetic crosses I've shown you, we've just used 1 letter to designate a mutant. Well, it's pretty obvious that if you're only using 1 letter and you have more than 26 mutants, you're in trouble. So the convention in all research fields, or most all research fields, is to use a 3-letter combination, which gives you much greater range and also gives you more expressivity. You can actually call something extrafloral organs, and it makes sense relative to the phenotype that you see rather than just calling it E. Well, what does E actually mean? So as we go forward in the course and we talk about different genes, we talk about mutant alleles of different genes, you're almost always going to see this 3-letter designation, sometimes a 2-letter designation, but usually it's 3 letters. So lowercase refers to the mutant version, uppercase refers to the wild type. But lowercase refers to the mutant. It doesn't necessarily say whether that mutant is a dominant allele or that mutant is a recessive allele. So that's what we're asking with this cross. So our parental generation is EFO homozygous mutant crossed to wild type, which is homozygous wild type at that locus. It makes the product that is lacking in the EFO mutant. So the F1 progeny of this cross are all going to be heterozygous. They're going to have 1 normal functioning copy of EFO, and they're going to have 1 non-functional copy of EFO. Now we allow these plants to self-fertilize, or actually we can look at the F1. If the EFO heterozygous plants in this F1 have normal flowers that look like the wild type parent, then the mutation has to be recessive, right? Because you have 1 functioning copy and 1 non-functioning copy. So this functioning copy allows you to make 4 flower organs as you're supposed to. But there's another possible outcome, which is that these heterozygous plants could have mutant flowers, like this one. In that case, the mutation is dominant, right? Because the definition of dominant is a phenotype that's visible even when there's only 1 copy. If that's the case, somehow this mutant EFO product is interfering with the activity of the wild type product and not allowing it to make the normal 4 flowers. Okay, so this is a very simple way that we can take a new mutant and identify whether it's dominant or recessive. Yeah?
Could you use heterozygous just as well as using a homozygous, and then just see what kind of, like if all regular flowers comes out even with the heterozygote? I guess if it was a heterozygote and it was showing not normal flowers, I don't know. Just because the thought to me of using homozygous to prove that it's homozygous recessive, to prove that it's recessive by using homozygous recessive is a little bootstrappy. So I was trying to, if you can use the heterozygous one.
Well, here we just know it's homozygous. We don't know whether it's recessive or not.
Yeah, but because if you're saying you're using the homozygous recessive one, it's just for me, I want to think you can use the heterozygous to prove it as well.
You could, but you can't. You'd have to go to the F2 generation. Yeah, but certainly, yeah, you can do that Punnett square, and you can see what the assortment of alleles would be. And yes, you could use a heterozygote. Yeah. But it's just more straightforward to use the homozygote. So the last example is where you've taken your new EFO mutant, and like any good scientist, you want to find out if other genes or mutants have been identified that look like this in the literature. So you do a search, and sure enough, you find that there's another mutant that's already been identified, and it's called CLV1, and it looks very similar to what you've isolated. So is it a mutation in the same gene as CLV1, or is it a mutation in a different gene that just affects the same trait? You can answer this question, again, using genetics. In this case, you're going to take your homozygous EFO plant and cross it to a homozygous clavata1 plant. And now, we're going to make the assumption that these are both homozygous recessive. Okay, just for the case of simplicity. They could be homozygous dominant, and you could also do the analysis, but it's a little more complicated. So we're going to assume these are both homozygous recessive. And we're going to look at the F1 of the progeny. And again, there could be two possible outcomes. One outcome is that all of the F1 flowers are mutant and look like these. The other outcome is that all of them are normal and look like wild type. Well, how did that come about? Let's look at these two outcomes in more detail. The simpler one is where you have 100% mutant flowers. And what I've done to illustrate how we get 100% mutant flowers from this cross is I'm showing each of the EFO alleles. Okay, these are not chromosomes. This is just one gene in blue. And somewhere in this gene is a mutation that knocks out its activity. So homozygous recessive EFO plants have two copies of the gene, and they're both defective. So we're going to cross this plant to plants that are homozygous for the clavata1 mutation. If you get 100% mutant flowers, it means that the clavata1 mutation was in the same gene, someplace else on the gene, but it also knocks out the function of this blue gene. So in the progeny of the cross, the progeny are going to get one copy of the EFO defect that inactivates the gene up here somewhere, and one copy of the clavata1 defect, which inactivates the gene down there somewhere. So you have two copies of the same gene, and they're both defective. So that's how you get mutant flowers. You're never going to get functional wild-type flowers out of that cross. Does that make sense? All right. But if you get 100% normal flowers, it's because there were two genes involved that both affect the same trait. In this case, you have gene 1, blue gene, which is mutated in both EFO alleles. But you've got another gene, the clavata1 gene, in green, which is normal. In the clavata1 mutants, that clavata1 gene is knocked out, but the EFO gene is normal. So when you make the cross, in the gametes, you're going to get one defective blue copy, one normal green copy from EFO. From clavata1, you're going to get one normal blue copy, one defective green copy. So in the F1 plants, you're going to have one normal copy of EFO, conferring a wild-type EFO phenotype. You're also going to have one wild-type copy of clavata1, producing a wild-type clavata1 phenotype. Wild-type was wild-type. One copy of each of the genes is normal. You get normal flowers back. Okay, so this is a very, very important application of genetic techniques in a research situation to find out whether the gene, the mutant that you're interested in, is something that's already being studied by something else, or if you've got something novel and you want to do your PhD project on it. So are there any further questions on what we've gone through today? I know it's a lot of material to take in, but I'm sure Brad can help you if you have any detailed questions. I'd be happy to answer anything at this point. Yeah?
Just out of curiosity,
Uh-huh.
was it recessive or dominant?
I'm sorry, out of curiosity,
Was it recessive or dominant?
the EFO mutant, that one was recessive. Yeah. Yeah. Anything else? Okay, that'll do it. Enjoy your weekend.